Okay, good morning, everybody. Happy Friday. Uh, we got super close to getting through chapter 9. I feel like probably halfway, really. Uh, so I know I left off thinking a little bit about some differences that we can see, and we talked a little bit about like some biochemical tests. We're going to see some more stuff as we move through. Really thinking about different potentials that microbes may have to deal with maybe an adverse type of an environment, dealing with different substances that might be toxic or flat out are toxic to cells that give them a leg up. So some, maybe many organisms have the enzyme catalase. You did a bit of this in the lab and looked at a few bacteria. Really, when I think about catalase tests in a lab situation, the big idea for a catalase test is to differentiate between staphylococci and streptococci. There are boatloads of other players that we may see as catalase positives, but when it comes to those two, this is a pretty quick mechanism to differentiate between the two. You maybe have the hint by some gram staining as far as who these potentials are, but a quick catalase test can definitely give you some confirmation. So staphs, aureus, epidermidis, and a boatload of other players are catalase positive and have the enzyme catalase, which gives them the opportunity in the presence of peroxide, thinking maybe like some of our immune cells having these capabilities to drill organisms. They're like, whatever, I drink that for breakfast. They take in the peroxide. The enzyme splits the hydrogen peroxide molecule apart, generating water and oxygen gas. That's the bubbles you see as the positive result, oxygen that's being evolved through that enzymatic reaction. Some have it, some don't. And like I said, with bacteria, even our own cells, our blood cells, we can see catalase activity. That's a different story, same idea. But a very good mechanism, thinking those gram-positive cocci, the streps and the staphs. Lots of others, a lot of gram-negative show catalase activity. There's a lot of other opportunities to see that. But then if you were to look at that and recognize virtually all of these guys are doing it, that wouldn't be a test that you would use really for any sort of differentiation. It's those positive cocci that we think of as more of a importance. All right. There are also, and I don't think you guys are going to work with blood agar plates, but these are a type of media that is very similar to your nutrient agar plates, an enrichment plate that lets lots of stuff grow, but they also can differentiate between a handful of different potentials for how those organisms might be utilizing the red blood cells. I know we talked a little bit about this stuff previously. So in this image, we've got two different types of bacteria that are plated on these separate plates. And you can see the one here with the beta hemolysis. This one's a good one to show around where this zone of hemolysis is, the bright red plate. This is what it would look like before you ever put anybody onto it. Very opaque. You can't see through it. You shouldn't be able to. These plates, various options for what type of blood. Oftentimes it's sheep blood. When we're thinking about Streptococcus pyogenes, that's a very good one to utilize. But there's other stuff. You can use cow blood, whatever. These plates, the agar gets prepared. It's got nutrients in there to help the bacteria grow, but the blood is providing the bulk of those nutrients, and it gets added after the media has been cooked, brought down to about 50 degrees Celsius. It aseptically gets added to those plates, about a 5% solution, and then the plates solidify. All right. So when we plate out the bacteria, and this would be like some streaking for isolation, although up here it looks like it's maybe not. We always want to try to streak for isolation. All right. We've got two different types of bacteria. There are those that we call alpha hemolytic, and they like the red blood cells, but they can't fully destroy them. They partially lyse them. So you get kind of a tannish appearance. It's slightly broken down, but I wouldn't call it see-through. All right. You can also see a few little bits in here where the inoculating loop has been poked into the media. Sometimes we can enhance the growth of some of these organisms, giving them a slightly anaerobic situation. And we also can see differences with their hemolysins, their enzymes to break down these cells, when we poke it into the media to get a little bit of a better perspective. All right. So I'm pretty sure the text talks about who these players are. I don't remember that off the top of my head. I'm going to make it up because that's all fine and good. All right. What I would like to imagine is we're looking at two different streptococci. Streptococcus pneumoniae is alpha hemolytic, and Streptococcus pyogenes is beta hemolytic. And it completely lyses these cells. And you can see over here where these have been inoculated, and truly it's the stabbing into the media. I can see 4 spots where the loop is poked in there, right? There's a big zone. And if you were to take, oftentimes when we use these blood auger plates, we hold them up so we can see the light, and we're looking at the bottom of the plate. Can you see through it completely or not? All right. This is a really pretty good one with the lysis, and it's not even being held up, and you can see that it looks a lot just like nutrient auger now. All right. So this is also something that becomes important when we start thinking about the potentials for some of these microbes to be disease causing in the first place. We may call some of these hemolysins, the ability to break down these cells with these enzymes, we may call these things virulence factors, something that enhances the capacity for these bacteria to truly establish and start causing disease. If they can break down your red cells and start getting all of these nutrients, the potential for them to grow very well and establish a bigger concern, the actual disease state, is there. And as thinking strep throat and seeing what this is doing to the red cells, that also makes you think a little bit about why strep throat truly is so painful, as it's trying to get in there and feed. You get a lot of inflammation, a lot of redness and pus. We'll talk more details about that stuff a little bit later. All right. So I know you inoculated Sims Deeps. They have the potential to show you motility. The M in Sim is for motility. All right. Sulfur, indole, and motility are what those letters stand for in those tubes. All right. That can work as long as you don't have an organism that's capable of breaking down that sulfur, because if it does that, the whole tube ends up turning black, and you can't tell if that organism's motile or not, right? So there are other tubes that we might use, especially if your Sims turned black and you're still wondering, are these suckers motile or not? So you could use just a deep tube. So instead of the slants, like you're probably pretty familiar with, this auger as it is put into the tubes, cooked up, and then allowed to solidify, it's solidified upright in the rack. When they make your slants, they cook them up and they put those racks on a little angled table that just lets them solidify at those angles. All right. Giving you a slant. All right. So there's a lot of different potentials with this. It could be a nutrient auger. It could also be a gelatin instead of auger that's in there. And that would also give you a potential to see if they've got gelatinase, if they can hydrolyze the gelatin and liquefy it, turning it back into a goo instead of a solid. All right. Either of those tubes, if you stab it in there with the inoculating needle, poke it right in and pull it right back out, as you incubate it, they will spread out and look, you can kind of see it here. You can see this cloudiness that looks kind of like a wedge as it goes in to the media, spreading out more toward the top and then narrowing down. All right. And over here in the negative, you can kind of see where the needle stick went in. All right. You can also, and this is microbiology, sometimes you got to use your imagination. All right. You can see right here where my arrow is at, the littlest bit of growth. All right. The bacteria is growing, but it's not spreading out. It's just right where the needle poked in. All right. Not motile. And then spreading out, motile. All right. Your Sims, have you seen the Sims results? Did you see some stuff that looked like that? A little bit of motility potentially. Did you see some black ones? Okay. You saw what you needed. And did you add the Kovacs and do the indole test? Stinky stuff. All right. Good times. So you also potentially can see some details with this where maybe you just get a bunch of growth right on the very top and nothing like we see over here, a little bit growing in there. All right. Well, would that tell you? Oh man, that ought to be on the test. What do you think if it's just growing right at the top? What would we think about its oxygen needs? Or it's aerobic, maybe obligate, an obligate aerobe. That sucker's got to stay right at the top, right? It can't grow down here where oxygen's a little bit limited, right? I think of these guys, non-motile and micro aerophilic, right? Or potentially facultative. They're still growing down there, but not an obligate aerobe, right? So you can get a handful of things by stabbing this in there, more than just like, is this sucker motile, right? And dependent upon your tube, you also could figure something else out, like maybe it can break down gelatin, right? Or it can't. So you will, I believe, do some motility tests. You'll have some nutrient tubes that you'll stab into and see some of that as well. Sims can give you similar details. All right. I personally, doing those biochemical tests, the Sims tubes are some of my favorite. I think it's really pretty cool to see all of the different things happening in there, and the ones that turn black, I'm just like, yes, bad news, right? Is oftentimes what I think about with some of those. All right. Doesn't have to be. So we are thinking about all sorts of different potentials for microbes and their capabilities to survive. Oftentimes when we think about most bacteria, they like to be pretty close to neutral when we think pH. All right. Little bit of deviation one way or the other. All right. Most of them fit into those categories. All right. But then there are some that are more acid loving or more basic loving. There's definitely some extremes when we think about some of these potentials. So looking at this, all sorts of different food stuffs that maybe you like to consume. How many sauerkraut fans? Yes. Kimchi? Oh, do you like kimchi but not sauerkraut? Okay. Because you want the spice. Okay. Okay. Have you tried sauerkraut and you're just like, that's gross?
No.
Why not? It's the same stinking thing without the spice. Okay. Feels different. All right. And homemade sauerkraut and store-bought can be very different. Not that I'm saying you ought to make your own sauerkraut, but you're in microbiology. I think you could. I think you should. All right. All you need is a little cabbage, a bit of salt. You get it in there and you just put a weight on the top of it and let it sit. And there you go. Just on your countertop. And that sucker starts fermenting all of the bacteria that's on the cabbage in the first place. It's already there. This bacteria, given the nice environment, it's got a little bit of water in there too. Just a little. And as it's sitting there, and typically you've got like a plate over your bowl that it's in to keep most of the air and most of the cabbage not exposed to any of that. And then something like a quart jar full of water pushing down on that plate. And after a little bit of time, a week or so, your cabbage has got very soft, the maybe slimy. It's got very soft. The lactic acid bacteria have started to break it down and ferment, giving you the nice tang to it. You throw some spices in there, some peppers, and then you get your kimchi. Same idea. It's just flavored very differently. All right. Potentially very spicy stuff. Just depends on your tolerance, I suppose. Pickles, yogurt. We've got some pico de gallo over here, which in this case, I don't know, not necessarily really bacteria dealing with this. It's lime or vinegar, but you ought to be putting lime in there to give it the acidity. All right. And we think about this not only in production of stuff, but we could also think preservation, like our pickles over here. All right. I think those are pickles. They're pickle slices, right? Which aren't the best usually. I don't know. You could get some spicy ones. All right. So, oh, and look at this red cabbage with apples. I've never heard of such a thing. That sounds delicious. All right. I'll have to go to the store and see if I can find them. All right. You never know. All right. And yogurt, there's so many different lactic acid bacteria. If you look at the label, lots of potentials. I don't know if they still, I think it's Activia maybe. They used to say all sorts of stuff about it being super good for gut regularity. And my thought is it any different than any other yogurt? I mean, yogurt's yogurt, isn't it? Maybe, right? They claim to have more bacteria in their yogurt than other yogurts. How would you know if they had more bacteria? It's a good question, isn't it? Makes you wonder if paying extra money for that Activia is actually worth it. If you're taking it either because you can't poop or you're pooping too much, one way or the other, you're not regular, right? So you're like, I gotta eat some yogurt, right? Do you think that just because they say it's going to help you do your business is any different than Fit and Fruity or whoever else they've got, right? It makes me wonder. All right. They claim to have this bacterium called Bifidus regularis. And with a name like regularis, you're like, sweet, that's going to make me poop regularly, right? That's what I need, right? Bifidus regularis isn't even its real name, right? They've trademarked that as a sales tactic, all right? So now you're like, wait a minute, what are they truly trying to do, all right? I believe it's Bifidus animalicus is who that truly is. I believe. All right. I did a little digging many years back because I had curiosity about this stuff. I'm like, what is really going on with this? I never got to the bottom of is it truly different? Because deep down, I think it's not. All right. Other yogurts oftentimes say bifidus, and you will see other things like Lactobacillus acidophilus, but you just see bifidus. They just give you the genus, not a species name. Why not? Part of me is like, it's because Activia trademarked Regularis, and they're like, we've got the same thing, but we can't say it, right? It's been trademarked. I don't know if that's true or not, right? Oh, and I've got somebody chatting right here. I better see what's up. So, both what? Okay. It's hard to say. All right. I don't want to turn you away from Activia if you truly feel like that stuff works for you, but it makes me wonder, is it all really the same? I don't know. I sense a pack back about that. Someone's got to do some research about Activia and try to truly get to the bottom of this, right? Uh, Spark some interest for me. I'll spark it. All right, we will see. Oh, sauerkraut and kimchi. All right, there's chatting going on in there. Both super tasty. All right. I've never made kimchi, but I have a friend that has made it. I've made sauerkraut. We made it in microbiology lab when I was a student, and we were going to consume it. And then as we pulled all of our stuff off, there was a little bit of fungal growth in there, which technically you might get a little bit of stuff if you're doing it at your house. You just skim it off and you just let it keep going. Uh, our professor was like, nope. And all of that went away, which is truly probably for the best. I don't think any of us would be like, oh, sweet, moldy sauerkraut, right? Somebody out there's like, that's my favorite, not me. All right. So this goes to kind of what I was hitting at with the pH. Most of them that we really think about, and a lot of them that we deal with in the lab, are in this green zone right here, what we would call neutrophiles. They like it pretty close to 7. All right. They can tolerate a bit of extremes, if you will, but not something super extreme. So 5.5-ish to 8.5. But as we look at these figures, they're a lot happier peeking out up here at all of these tops of our humps. This is their sweet spot, if you will. So right around neutrality is where you get the best growth with our neutrophiles. Can they grow at some altered pHs? Yeah, but not as good. All right. The acidophiles, our blue ones over here, grow optimally around a pH of 3, but they can tolerate very acidic environments, pushing toward 1 and going toward neutrality. They're still like, it's okay. It's not the best. All right. And you can see them to about 5.5 as well. All right. There are some archaea super extremophiles that push this threshold even farther, growing toward a pH of nothing, right? Right around 0. That's super crazy, all right? Or the other extreme, pushing toward 14, and we're not even going that far out here, all right? But with our bacteria, we don't think about them pushing that threshold either way. The alkophiles, basic stuff is what they like. pH close to 10 is their optimal, but they can live okay, not the best, towards neutrality, and then the same as we push it toward more basic values. This was a lab, I'm pretty sure we did not bring it back. This was a lab that was done all of the time in micro labs. And there were a couple of others, like maybe some salt. there were multiple tubes at all varying pHs, broths, and you would have a couple of bacteria that you would inoculate into every pH range. So if you had two bacteria you were testing and we have 5 different pH ranges, you got 10 pH tubes. And that's all, that's just the first thing you're doing. You got a boatload of other things. That's a lot of stuff to be inoculating. And then you have to stop and think, there's 140 people in this lab this semester. And if we're doing this stuff in pairs to minimize it, that's still a boatload of tubes. And the stockroom is making all of this stuff. And when it comes to the pH and salt lab, they're just like, oh no, right? Opening new cases of test tubes because we got to use so many of them, right? As soon as we decided maybe we don't want this, you guys were happy. You don't even know you're happy, but you are. You're happy about it. The stockroom was super happy about it. And deep down, really what you would see, too acidic or too basic, our bacteria that we work with flat out don't like it. There's a couple of them that you could see as kind of acidophilic, but it really wasn't anything that was like, oh, that was a great lab. It was pretty much a waste of time inoculating a bunch of tubes to see that this crap didn't grow. You need the right pH, and salt kills too, right? If you've got something that's flat out too salty, unless you're Staph aureus, you probably aren't going to be growing in there. It was kind of interesting to see that you could get some of these growing in these super saline environments, but ultimately most of the other players you put in there just flat out don't grow, right? So both of those we kind of thought sort of just a waste of time to really see that I got nothing, right? And I feel like a quick spout about it in here probably sufficient. All right. This isn't the Great Salt Lake. You're going to see it in 1 second. This is Lake Natron, Lake Salt, in Tanzania. And we can see a lot. This is an aerial shot. It almost looks like you're looking at a petri dish with a bunch of weird crap on it. But you can see the lake and a lot of pinkish red within there that are the result of some of these halophilic microbes, salt lovers. And my gut says halobacterium, which is an archaea, all right, is likely who we see over here. This is also a lake that is super basic. So we've got alkaphiles and halophiles, maybe both in the same organism, that thrive in this habitat, all right? And it also looks like some pretty big just salt deposits that we're seeing out there. I'm thinking salt islands, but I don't know, all right? That's kind of what I'm thinking from this aerial shot, maybe, all right? So I'll show you Salt Lake in just 1 second. There are other ideas about some of these microbes that the black smokers that we're seeing here in the bottom of the ocean by some of these hydrothermal vents have opened up our eyes to like a completely, what I think of really as kind of an alien world, all sorts of weird stuff happening down there and thriving in this habitat that is super inhospitable. Very high temperatures, couple 100 degrees Celsius, super high pressure as we are deep down in the bottom of the ocean, massive amounts of pressure. How the heck can you survive in those types of habitats? The barophiles are bacteria or archaea that can tolerate high pressures, right? And then the thermophiles or hyperthermophiles are the bacteria or archaea that can tolerate super high temperatures. And in some instances, high pressure and temperature together makes it a little more stable for these organisms. So a bunch of nutrients spewing out through these black smokers, sulfurs that a lot of these organisms are potentially able to utilize. We may describe them a bit later as a chemolithotroph using some other molecules as sources of electron acception or energy. They in turn feed upon this. You get a big bloom, if you will, of these microbes. And then we get all sorts of other players that now have foodstuffs to be consuming. So these bacteria get fed upon by different microbes in the water. And also we can see all of this reddish stuff. These are tube worms that are flourishing on this mound. They like the temperatures and they like all of these microbes, and that's what they are consuming. So they're flat out living right at the buffet, all right? The best place to be, right? Eat a little bit, take a nap, wake up, go back to the salad bar, eat a little bit more, take another nap, right? It's all you can eat. That means you should stay there all day, right? Probably. All right. So then the question really comes to my mind, how can you get organisms from this type of a habitat and bring them back to a lab and expect that you could cultivate them in the first place? Where are you going to grow them? An autoclave, right? There's some serious issues with trying to deal with this. So you can see we've got some devices down here that are making some measurements. We are doing this in situ, looking at this stuff rather than trying to collect it and bring it back to the lab and try to plate it out in atmospheric conditions instead of super high pressures, right? They wouldn't survive, all right? So there are potentials to get some of this, just dependent upon how you are dealing with them. But a lot of it is making measurements in place and trying to detect what do we have going on with different molecules and microbes within that. So this gives us the temperature range. And again, for who we think about for the majority of stuff in here are these guys, the mesophiles. These are the organisms that like temperatures that we like. So body temperature 37, optimal for these mesophiles. They like the same stuff. So when we think about disease-causing organisms, this is really what we're thinking about. The mesophiles are the ones that are the problems, all right? Then as we look at it, we've got other possibilities. Sacrophiles are cold lovers, and they prefer more like refrigeration temperatures. They can survive below 0 degrees Celsius. Not the best, but they can still be viable. And they can also get around room temperature-ish and still have potentials. Again, not super happy, but capable, all right? So you can see there's a little bit of overlap. And there are some potentials for some microbes to maybe be a sacrophile or a sacrotroph. So it may be more mesophilic, but it's like, you know, the fridge is still fine. It's a little bit chilly in here, but I can still tolerate it. Staph aureus fits into that category as being sacrotrophic. It can still be hanging out in the fridge that we oftentimes think about as a mechanism to inhibit the growth. It's too stinking cold, right? Staph aureus is like, nah, it's fine, right? This nice custard dessert that you made, maybe it's got a little staph in there, then it goes to the fridge, and then you get ill as you pull it out and consume it. Custards and creams, things like that, are pretty good for some foodborne illness if it's not handled properly. And if you didn't wash your hands as you're making that stuff and getting into it, right? That's an issue, all right? there's some other stuff like listeria that fits in this category as well. It likes the fridge. Lunch meats and soft cheeses can be some potentials with that. I know there was an ice cream recall just a little bit ago, last week, 2 weeks ago, something like that. And listeria was one of them. That's freezer temperatures, right? It's okay. We're okay. That's not so good as you're getting your bowl of ice cream. What the heck can you eat? Kimchi, right? All right. The thermophiles, we see these guys over here in red fittingly because they like the heat, right? So from temperatures toward the end of our extreme for the mesophiles, 40-ish, but they like it around 70. And then towards 80, they start dropping off. The hyperthermophiles like it even hotter. These are things we're thinking like the last image down at the bottom of the ocean, above boiling temperatures, and still potentials, right? And it's things like this, probably somewhere in between here, that set the stage with like the PCR stuff, our Thermus aquaticus growing in the hot pools in Yellowstone, that lent to some ideas, because typically these temperatures denature proteins, they inactivate enzymes, but thermophiles and hyperthermophiles, those types of enzymes, their proteins must be stable at considerably higher temperatures that our proteins would be breaking down at, right? What's up with that? Could we use that? Obviously so. Okay, so this, another aerial photo, we're looking down at Lake Erie and we could see all this stuff that I think is really kind of pretty looking, but then as you think about what it really is, it's disturbing. So here we could see the shore and a bunch of fields that we're looking at. I'm assuming maybe a bit of residential stuff right there, but this looks like a bunch of farmers' fields, checkerboard sort of stuff, and fertilizers getting into the lake and the eutrophication. So we've got a huge algal bloom that looks like it's swirling and churning around in here. And truly the result of seeing this big burst of algae is the result of a lot of runoff from all of this agriculture button up right against the lake. This image is, I think, interesting, but I also think a bit disturbing. So I told you we'd see Salt Lake. This is another aerial shot. We're looking at the northern portion of Salt Lake, and we're looking at a hint of the southern portion of the Salt Lake. All right. And the southern portion is definitely considerably larger than the northern portion. And it's both sides. We're seeing it shrinking with droughts that Utah is experiencing. I'm curious what it looks like now. Probably a lot less going on up here. All right. So a little bit of a story with this. You can see a nice line splitting this lake in half. All right. Many years ago, the Army Corps of Engineers put a railroad track through the lake, cutting it in half. They built an earthen dam going through the lake, and there's no coverts, although there are talks of correcting this decades later. There's no intermingling from the northern portion to the southern portion of the lake. They are two separate lakes, all right? And with that, there's some issues. There's some differences, all right? There's not as much water, fresh water, coming into the northern portion as there is in the southern portion, all right? And in both portions of it, a lot of the water, especially in the northern portion, a lot of that water is diverted for agriculture and not allowed to go into the lake. So we're already cutting off some of what should be going in there. And it sets the stage for the top bit to evaporate more, to concentrate the salts. And these halobacteria in the northern end are like, sweet, sweet, I finally have the buffet. They are super happy with those salt concentrations and they bloom. All right. So you get a two-toned lake, especially in the summer months, that are visible. You can see it if you go there. You can see it if you fly over it. The astronauts can see it from the space station as they look down. That's cool. All right. They have satellite images. If you watch the weather, they show pictures, and it's definitely more apparent in the summertime when you see the satellite image, two-toned lake. All right. Pretty interesting. All right. And dependent upon when you see it, you might see blotchiness with some bigger pink blooms and more kind of this, because it's not kind of fully distributed yet. So there are big pushes to try to mitigate these issues by not removing the train tracks, but by removing part of that earthen dam and putting coverts underneath to allow for the exchange, to allow for some intermingling. And yeah, whether or not that actually happens anytime soon, we will see. All right. Have you guys worked with McConkey's auger yet? EMB auger? No selective or differential media yet. Okay. So these types of plates, you're going to see a handful of different things coming up, I'm assuming, in the near future. Mannitol salt augers, McConkey's, or EMB plates. These types of plates are what we call selective and differential. They have nutrients in there to enhance the growth of some, and they have other substances to inhibit the growth of others. We use these plates to weed out some organisms, and we also, with the differential stuff, use them to tell some apart. Some that may use something or not. All right. So this McConkey's auger, McConkey's and EMB, EMB stands for eosine methylene blue. Both of those types of media are used to grow gram-negative bacteria, and they also have some stuff that helps us to differentiate. They contain lactose in the media, and I know you did those phenol red sugar ferments, and you had a lactose tube, right? That lactose, if it used the lactose, it turned it yellow, right? Showing that it was able to break it down, and it fermented it, making the pH lower, and you get a yellow color in that media. The McConkey's plates, it's got lactose, but it's different as far as how those colonies appear. So we've got two different bacteria. E. coli is this guy that's bright pink. If that organism, whoever it is, E. coli in this place, if they can use lactose, when they're pulling it in, their colonies turn pink, and that's an indicator that they fermented the lactose. Up above is a different bacteria, also gram-negative. It can grow on the plate, but it doesn't use the lactose. So their colonies are clear, all right? Kind of cream colored, definitely not the pink, all right? This could be Pseudomonas as a potential, all right? It doesn't ferment lactose, and E. coli does, all right? I said both gram negatives. We inhibit the gram positives in these plates because crystal violet, the stain we use in gram staining, crystal violet is incorporated into this media. And gram positives, when they're trying to grow on this plate, they're pulling in the nutrients, they're pulling in the crystal violet. And the crystal violet in a gram positive with a super thick cell wall, it gets into these cells, and as they're trying to replicate, they can't make their peptidoglycan layer because it's all getting crystal violet rammed up in there. They can't replicate, so they die. The crystal violet inhibits their growth, right? But the gram negatives, with their outer membrane, crystal violet does nothing to them on these plates, and they're able to proliferate, all right? So it's selective by that crystal violet, and it's differential by the lactose that allows us to tell the difference between these two gram negatives, all right? The mannitol salt agar plate allows gram positives to grow, but truly those, since salt is in the name, those that are more capable of growing in that situation, like staphylococci, all right? So it's not the best to separate out, all sorts of gram positives, but to differentiate some of the staphs, it works very well, all right? There are some others that you may use as a weeding out plate. We call them P plates, not urine, all right? P for phenyl ethyl alcohol agar. And these alcohol plates do the opposite with our gram negatives. Just as the crystal violets inhibiting those positives, alcohol deteriorates the outer membrane of a gram negative. Now those suckers die. They can't tolerate those plates, and your gram positives can grow, all right? There is a little bit of wiggle room because some of them, some gram negatives are like, you know what? I like a little alcohol myself. I'm gonna tolerate it. They don't grow the best, but they still grow a little bit. So that can make it a little bit of an issue. All right. So I mentioned the MSA. Here we've got a plate. We see gram positive cocci in clusters. My gut says probably some staphylococcus. All right. If I had this in a slant or on a nutrient agar plate, I could take a bit of that colony off and slap it onto a slide, put a drop of peroxide on it and see it bubble. And I'm like, okay, now I know. I'm looking at staph. All right. But who is it? Is it aureus or is it epidermidis? Oh, I want that plate up here. An MSA plate, I thought it was coming up. I think we'll talk selective and differential in a bit, but I'm already spouting it off. An MSA plate, if you had your staph organisms, and I said, who is it? Could it be aureus? Could it be epidermidis? For sure. Could it be somebody else? Probably. But these are the two we're thinking about, right? Both of them would be catalase positive. Both of them look like this under a gram stain, but we could tell them apart with an MSA plate. And Staph aureus grows on this plate, and it also likes the sugar mannitol. It's a sugar alcohol with an all on the end. You should think alcohol. So mannitol, sugar alcohol, Staph aureus likes it, and it ferments it, and it turns the media yellow. All right. So now you've got a plate that's turned yellow. You've seen these gram stain. You've maybe seen the bubbles. You got Staph aureus, right? Staph epidermidis can't ferment the mannitol, but it grows, it likes the salt. It grows on there. They're little white pinpoint colonies on an MSA plate. Saw the same thing in a gram stain. Now you know, all right? These guys should be epidermidis. Didn't ferment anything. My other ones should be aureus. There you go, right? Unknowns coming up. You'll see some of this stuff. All right. That's the end of that. As I see exercise, I know it's time, all right? Time for exercise? No. Time for questions about these tubes? I think not. All right. I will stop at that. Unless you want to spout off, who's this guy? Nobody. I knew it. All right. Put that on the test. I bet you that's an obligate anaerobe, right? He's growing clear down at the bottom, right? All right. Enough of that. Farewell.